So I'll introduce Oliver. So I'm delighted to introduce Professor Oliver Fiehn from the University of California, Davis, West Coast Metabolomics Center. The title of his talk today is going to be How Many Lipids Are Unknown? Large-Scale Lipidomics Cloud-Based Data Processing with LC-Binbase. So thank you everyone for joining us today. My name is Valerie O'Donnell. I'm based at Cardiff University in the UK. As usual, the webinar is being recorded and it'll be uploaded onto the Lipid Maps YouTube channel. Will be about 45 minutes with about 10 minutes for questions at the end. So if you could go ahead and put your questions into the chat or the Q&A during the talk, that would be great, because what I can do then is identify people asking questions, and then I can hand the microphone over to you and you can ask the questions yourself. You can unmute and ask your questions direct to Oliver. So please post questions during the talk. So I'm going to introduce Oliver. I'm delighted to introduce you today, Oliver Fiehn from UC Davis. He's pioneered developments and applications in metabolomics with over 400 publications. He started out in '98 as a postdoctoral scholar, and then from 2000 onwards as a group lead at the Max Planck Institute for Molecular Plant Physiology in Potsdam, Germany. In 2004, he became professor at UC Davis Genome Center, overseeing his research laboratory and also the core service laboratory for metabolomics research. In 2012, he became director of the UC Davis West Coast Metabolomics Center, currently supervising around 35 staff and operating an enviable number of 18 mass spectrometers. That's pretty impressive. Since 2021, he is directing activities at the new Thermo Fisher Center of Excellence in Clinical Metabolomics at UC Davis Clinical Campus in Sacramento, California. Oliver is lead on several NIH-funded projects, including dietary metabolomics, the Longevity Consortium, and acute to chronic pain. And alongside this, his laboratory builds software and databases hugely widely used in the mass spectrometry field, not just for metabolomics, but also for lipidomics. This includes MassBank, hosting around 680,000 public metabolite mass spectra, in addition to the really widely used, well-known software MS-DIAL, which is data processing software that he initially pioneered. And along with this, Investigate public front-end, hosting over 2,800 GC TOF MS metabolomic studies. Oliver co-founded the Metabolomics Society in 2005, and in 2017 he co-founded the Metabolomics Association of North America. He's won numerous awards for his research, including the 2014 Molecular and Cellular Proteomics Lecture Award and the 2014 Metabolomics Society Lifetime Achievement Award. So with that, I'll hand you over to Oliver. Thank you for coming today. Looking forward to hearing your talk.
Thank you so much, Valerie. It's great to be here and to e-meet you, so to say. So I'd like to present today a consortium effort that is the consortium of the NIH Metabolomics program. And in that program, we have worked for 10 years together, and the last 4 years with an emphasis on unknowns. So like what unknown metabolites are there, and of course, which unknown lipids are there. And we started actually with unknown lipids because we felt at least it's a little clearer in terms of the chemicals portfolio to look at. And the leader of this enterprise was Charles Evans from University of Michigan, who's prominently displayed here. And from my own lab, Dr. Tong Shen, she is a principal scientist in my lab, senior scientist, working alongside me. And then we had a couple of other laboratories helping us. So when we look into what people usually publish, in terms of interlaboratory and interplatform lipidomics trials, like Thompson et al. In analytical chemistry, Bowden et al. In Journal Lipid Research, and Contempois in Scientific Reports, we see that people talk about identified lipids. So they say, we have 1,500 unique lipids, at least in one lab found, or 300 lipids with good agreement between targeted and untargeted, or excellent quantitation reproducibility. But the question really is, what is the dark matter? What are the things we don't talk about? And what is there to find? And at least, would we all agree on what to find? So before we have to talk about unknowns, we have to talk about knowns, obviously. What does make a lipid structurally identified? And, there's just the word identification or the word annotations have different meanings. So it's relatively easy with a accurate mass spectrometer to come up with a mass, let's say 773, and find a 184 product ion and say, well, that is phosphorylcholine, with the mass 773. But if we look at lipid classes, then there's 202 at least types of phosphorylcholines that would fit that bill. So once we know a little bit more about the bond type, these are acyls, we can say, well, maybe it has 35 carbons and one double bond. And we can go down and we find the acyl composition, especially when we go to negative mode. We might find, okay, so that's really 18 to 1 and 17 to 0, but we wouldn't know SN2 or SN3 positions. When we do know that, then we can make the slash. When we then know where the double bond is, we can say, well, it's the 9, maybe at the 9th carbon. And eventually we'd like to know, well, is it cis or trans? Most likely cis, but you don't know. So for going down this pyramid, there's a number of different software, where we get first the elemental formula, then we might get list of likely structures. We can do structure expansions, for example, in the MINE database. We can then limit things by either retention time prediction or by collision cross-section predictions using ion mobility. We can further limit substructures by hybrid searches, and eventually might need to do chemical derivatizations like ozone to find out double bond positions and so on. But in principle, lipidomics doesn't usually go that way. We usually don't have the time to go all the way down to this very end. And what I will talk about today, what I will tell annotated, either stays on the bond type or at the fatty acyl composition, because even the regioisomers are not that straightforward. So that is what I will talk about today in terms of annotation and identification. But I just want to make sure that people understand when we give this structure that still 12 enantiomer isomers that could be done. So first of all, classic lipidomics, usually it has an aim, like pancreatic cancer, I understand is the next lecture by Hopacek. And you would go, you extract your samples, you do your workflow, and then you publish. And usually after you have published, you might think about making your data available. And then you might go to sites like the Metabolomics Workbench in the United States or Metabolites in England, and publish that. But then it's often not exactly findable, accessible, interoperable, or reusable, because you might leave out a lot of details. And so we here in the NIH Metabolomics Workbench, try to do the opposite. So we didn't have a clear biological design, but rather we went the workflow, and the idea was just to make the data public. And once the data are public, people then can think about publishing those, snippets of it, and that's kind of a different way of thinking. First, make data public, and then give it to the world for finding biological ideas and initiatives. So that is in principle what my lab does. And in this specific study design, we had 4 or more labs. We used more than 4 types of samples that were shared across the labs. We used sets of identical internal standards. That is an enriched /1 lipidomics kit from Avanti Polar Lipids, but we added acyl carnitines and free fatty acids to that kit. We used at least 1 identical chromatography, but people were able to add more methods. We used both QTOFs and Orbitraps in positive and negative mode. We couldn't define the mass spectrometer because obviously people have different mass spectrometers. But we then funneled back into one stream of identical data processing. We gave people the opportunity to use their own workflow, but obviously nobody has time to do that. So then the question was, when we do that, we take the same samples from 4 or more labs, how well can we then compare the results? What are the total number and types of both identified and unknown compounds? And do we even agree? Do we even agree of when we look at mass spectra, what these mean? And what can we tell about unknowns? So the key questions that we had is, well, will we have a scenario A where we would have the different laboratories here in circles with dark colors, having a Venn diagram where basically we have a big intersections of the knowns, and we would have a big intersections of the unknowns, of course, with some variance, because this is just experimental errors or small differences that would mean that one lab would find certain things and the other lab doesn't. Or, could we end up in something like that, where we wouldn't agree a lot about the identified compounds or annotated compounds, as we call them. And in addition, each lab would find a plethora of other compounds that would not be detected by others. And then, when we have the data, can our data help facilitate future annotations in other studies? Can we keep the unknowns and say, well, these unknowns are interesting. These unknowns will have now an identifier and can be rediscovered in future studies because they are in a database. Even if we don't know what they are, they become known unknowns. And we keep them, and they have, a value now. And eventually, when they become very important, then we will do our best to kind of really go to the structure of those unknowns. Well, what's the current status? What did we do? So we had these types of samples. We first of all had 4 types of standardized plasma samples, all from the US National Institute of Standards and Technology, from type 1 diabetes, high triglyceride plasma, young African-American plasma, and the standard reference material 1950. In addition, we had commercial, samples, bovine liver extract, heart extract, porcine brain, and human skeletal muscle extract. So we had, a couple tissues and plasma. And then we used different mass spectrometers, and also different instruments, of course, like Thermo Vanquish, Water Acuity, Agilent 1290s, but also, Q Exactives or QTOFs, and, different strategies to go and get as many mass spectra as possible, with different types of iterative exclusions, or on pool QCs, or on split DDAs on plasma, with ramped collision energy. So it's a little different what people did. We didn't want to prescribe it because it's still a little bit in flux. What's the best way to go about it? So in principle now, we had on the MS1 end and on the HPLC end, so the column and the buffers were fixed, but the MS2 or the data-dependent MS were varied a little bit so that we get a plethora of mass spectra. We used internal standards. I think the total was 76 internal standards, and used 20 microliters of plasma and similar amounts of tissues. So this is how the chromatography would look like. So you would have, the classic, and this is positive mode here, the classic way how things are looking. You would have lyso-PCs and so on in this region, classic PCs and sphingomyelins in this region, triacylglycerides and ceramides in this region. And in principle, we see that the liver is more rich in certain aspects. The brain is more rich in this region, but the types of compound classes are comparable. If we look at the chromatography between laboratories, we see then again that, overall retention times fit nicely, but sometimes they don't. Although we use the same column and the same buffer and the same solvent compositions and the same flow rates, the absolute retention times can change based, for example, on column delays, on the different connections that you have from the solvents to the HPLCs and from the HPLCs to the mass spec, but also the type of UPLC you would use, might have different settings so that you actually get differences in retention time. So with these retention time differences, we overcome them by internal standards. This is the classic way to do it in gas chromatography, and why not do it in liquid chromatography? And it works great between the laboratories, very straight. You sometimes have one misannotation of a compound, and you clearly see it by having a different retention time. So okay, this is not right either in lab A or in lab B. And here the same, you would say, well, this is not correctly identified, so you leave it out. You can even now use the common protocol, the standard column and buffers, and compare it to your in-house methods to project basically the internal standards and align them so that you can even compare data sets that have been run on slightly different columns or buffers. So that's working fine. So when we then look into the data processing, we used MS style here. At that point is version 3.6. I think right now it's version 4.8. Just for the broad view, we said, okay, let's do a dot score similarity of 70%, regardless of retention time. Search the precursor mass within 10 millidalton. Don't think about adduct joining right now. Just like to have a broad first look and about the acquired MSMs in positive mode and the putative first list of annotated annotations between laboratories. And we see that in positive mode, we go from 700 to 1600 annotated compounds this way, and 5,000 to 3,500 raw MSMS spectra. And in addition, we would have, of course, negative mode, going from 300 to 900 annotated compounds and 3,000 to 1,500 raw MSMS spectra. So obviously then joining positive and negative modes to avoid double annotations is yet another task. Well, that is just the first broad look. Then the question is like, which do we have a winner in terms of mass spectrometers? And just as here, just by generic annotations, the number of carbons and double bonds in terms of overlapping annotations. And we see there's quite a difference between laboratories on that level. But the reason was, obviously, as I said before, that people use different MSMS strategies. So I don't think this image would reflect the differences in instruments, but rather differences in how these instruments were operated in terms of speed and depth of MSMS acquisitions. But we see that, of course, there is an agreement and there's also disagreements between labs. So we have to kind of join all that together and then see if in principle the labs would have agreed on the MS1 level, and then some labs would have acquired an MSMS on that compound and other labs would not. So with that data in hand, we had to come up with a workflow and a schema how to prioritize the number of unknowns. Like, what is now an unknown? Which ones do we want to keep? And which of those horrendous number of spectra do we have to discard as being inconsistent over time? So we eliminated all features that were not present at least 3 times in the blank, in at least one sample on at least one assay in one lab. So that's a fairly relaxed way of looking at it. People might say, oh no, you need to have a 10-fold background subtraction and it should be on 3 assays. That's fine, we can also do it later. But we didn't want to throw away too much at this point. Then, of course, we wanted to know, are there knowns or are there unknowns that are specific for, say, a certain tissue type or a specific plasma type? And then we reduced by saying, look, if we look in the heart, we will only keep things, even on if the MS1 level annotation is found at least 50% of the runs of all samples across all labs. So that's was a fairly, more strict maybe conservative thing. So, that we said, no, it has to be found relatively consistently across the laboratories. And then we can, in principle, sort more. We could ever by the intensity, for example, saying, okay, so now the more intense compounds are more interesting than others. And then we could select final features. And for human plasma, for example, 917 unknowns would meet these criteria for human plasma. So when we do that, that's the data set summary. We had, of course, on the MS1 level, a huge number of feature counts. These are MZRT features. But then that was reduced when we thought about that, like in the blank subtraction and the missingness. We needed to make sure that we usually find all the standards automatically. And we did. Most of the internal standards were found automatically. And then, we thought, like, how many compounds do we now in our total data set annotate? And that is 2,300 almost, with additional annotations, meaning that you'd say, well, it's in the region of the phosphatidylcholines and it has this mass. So you could have a more putative annotation, like, as I said before, PC 35:1, then you would add, of course, had a lot more annotations, but these are pretty dubious. So we really here focus on those annotations only, and also for the unknowns that have an MS-MS spectrum. When we look at that, these are, not surprisingly, the types of lipids we would annotate, starting with fats, over phosphatidylcholines, diacylglycerides in positive mode, but also plasmalogens or ether lipids in general, both on the PC level and the PE level, ceramides, hexazosyl ceramides, but also ethanolamides, and then the many other types of lipid classes. In principle, similarly in negative mode. So let's look into the overlap analysis. It's just now only positive mode electrospray for the different organs. So we would have annotations of unique compounds, for example, in the brain. So we had 862 unique lipids annotated in the brain. But when we think about, like, how many of those lipids were specific, in this Venn diagram, we see that 191 lipids were specific. So that it means that organ specific, we had 22% of the whole brain lipidome annotations in this way, only positive mode, that was not found, not even on the MS1 level in any of the other organs. And that's kind of interesting. What are these lipids and why didn't we know about them before? So that's fine. And so in positive mode total, we would have 1,500 annotated lipids, and we see the intersections of these lipidome prospects between the organs. The same now we can do for unknowns, right? So for unknowns, we had 1,685 in the brain. And again, we found 691, which was 41% of the brain lipidome was not found in other organs. And that's kind of interesting. That is a little bit surprising, because when you look at textbooks and databases, it kind of always looks similarly. And yes, we do know that bacteria have specific lipids. And yes, we do know that certain fungi have specific lipids. But did we really think that organs would have specific lipids? That's not so well established, I think. And it's kind of cool to see that, and also to see that there's a lot of unknowns that are specific for certain organs. And so we can now bring it together. So on the one hand, we have in the brain, for example, 22% of known lipids that are not found in the other organs. 41% unknowns that are not found in the other organs. And we see this, specificity across different organs. We also see now already that the percentage of unknowns over the knowns, the specific unknowns over the specific knowns, is always higher. And we also see that when we look at this common core, that's the center of this Venn diagram, that there are, of course, more identified compounds than unknowns. So we do not have many unknowns that are universally found in across organs, which makes sense. And the percentage is higher in the common core to have identified compounds. So in negative mode, it would look similarly. So again, we would see that first of all, in the common core, the percentage of knowns is higher than the unknowns, whereas per organ, the percentage of specific unknowns is higher in every single organ than the percentage of knowns. Secondly, what we see, the coverage. Now we can also talk about coverage. So the coverage of identifieds per organ is about 20%, 16 to 21% in the different organs in negative mode, where it is about a third of all MZRT features with MSMS are identified across organs in positive mode. So we have a higher identification rate in positive mode than in negative mode, which is kind of expected because many libraries are a little better established for positive mode than for negative mode. But it's also meaning that, for example, triacylglycerides, they just come up in positive mode. So yeah, you'll not find them in negative mode. All right, so let's look into the plasma types. So we had 4 plasma types: diabetics, high TG, healthy young African Americans, and the NIST. And in positive mode, again, we have, about a third of the coverage is identified. We have a much higher percentage in the common core because now, of course, we look at plasma versus plasma versus plasma versus plasma. And we still have more unknowns in these identified. It's slightly higher, not as dramatic, but we have a slightly higher percentage in specific unknowns for example, diabetic blood, specific unknowns in healthy African Americans, and so on, than in the knowns. In negative mode, it is similarly, but again, lower coverage in terms of plasma in negative mode compared to positive mode, as we can easily see here. Even higher percentage in the common core that is common for all the plasma types across board. So that's kind of nice. And now, of course, we want to know, what are these unknowns? Well, some of them are just trivial. They're not really unknowns. These are knowns, but they're not lipids. They're contaminants or artifacts, like phthalates. Some unknowns are found, but they are noisy and low abundant and don't have a lot of information, as we can see here. So these are trivial cases, either trivial because we already know them, or trivial because the information is so bad that we can never use it. Some unknowns are unknown because they are not well covered in databases, like oxidized lipids. And lipids can be oxidized, and they're easy to identify by MSMS, only they have a shift in precursor mass. So we would know this is a triacylglyceride that has an additional oxygen somewhere. Well, there is not a great annotation, but at least we know more than we did before. So that's easy. So then we found unknowns that are in the literature, but you have to dig deep. So first we thought, oh, we have found a new unknown. And we identified it, and then we dig the literature, and then 20 years ago, somebody talked about it, or 10 years ago, in some paper somewhere deep in the bottom of the ocean, somebody already talked about that lipid. So there are rare lipids, I would like to say, for example, acylated lipids or modified lyso-PCs and lyso-PEs that are modified. I don't want to talk about it yet because we haven't published it, but you can imagine there are some modifications on it. And then we used LC-Binbase. So we harmonized everything in a big database stream. And then we saw things that are just unexpected. So they are not unknown unknowns, but there are unexpected known compounds. For example, epoxide lipids. So here's a regular oxylipin. We didn't expect to see it because usually it's low abundant, but it's a very nice MS-MS spectrum, very clearly identified by both MS-MS and retention time, and a number of exposome compounds, drugs that people take, that usually are ignored because usually in lipidomics we only look for lipids, but things are co-extracted, even with fractionation. If they are lipophilic enough, we'll find it, and then we can annotate those too. And then, we have unknowns that are interesting because we can, the MS-MS spectrum has some similarity to known compounds. And this is using here the Global Natural Product Service visualization, where each node is a compound. If it has an annotation, then we know what it is. The color in those nodes is reflecting where it was found in plasma, heart, and muscle, for example, this lipid, but not in the brain. And then you would have an unknown that was only in muscle, but it's kind of similar to that one, right? And the similarity would mean it's similar, but it has an additional C4H8 somewhere, right? So we can bring these into networks. So this is triglyceride-centric network, or you can do the same for other lipid classes like hexazyl ceramide-focused networks, which are sometimes abundant in brain with some species from other tissues. As we can see here, these are all very dominant for brain, but often not found in other tissues. All right, so these are interesting unknowns, and quite a number of them, as you can see. So that helps too. So how do we make it now coherent in a big database? And for that, we established in the last 4 years, really, with 4 software engineers, Diego Pedrosa, Gerd Wobegut, Daniel Rundle, and Sajan Mehta, now Nolan Guzman. We basically use the same idea as we use for LC-Binbase. So I'm running LC-Binbase for 17 years now, hundreds of thousands of samples, always with the same mode, where we first detect all peaks, we then deconvolute things, we find the internal sets of internal standards, we calculate a reference index for retention time, and then we go sequentially from the most abundant to the least abundant, and see, always check if a certain MS-MS spectrum is already in the database or not. We also say it has to have minimal thresholds for purity and signal to noise. And if it fails on some level, we will discard it. So that is, in a way, how it's done. We have rewritten our MS dial code for cloud computing. Here we use Amazon Web Services, basically for speed, because otherwise you get hung up on software if you do it on PCs. And with that idea, you automatically add new entries. We call these entries a bin because it's kind of like funneling something using all the information you have, which is retention time, accurate mass, and MS-MS spectrum. And if it's new, then you add it, and then you might annotate it. So this is in principle how LC-Binbase works. So we have on the front end a study design database that basically informs about the study design, species, organ cell types, and also treatment cell disease groups, so that we can later on say a certain bin, a certain mass spectrum, retention time, accurate mass feature, if you like, is found in a certain organ, say the brain, but not in other organs, say the muscle. And in principle, we can do this also for species and so on. So like a big database, but on experimental level, not on literature. Then we use the Lambda from Amazon Cloud Services to basically do all the things that MSDAL does, from peak picking, deconvolution, retention time alignment, MSMS quality. And we use Mass Bank of North America for data annotations. So that we would have, in principle, 2 mass banks, if you like, one for knowns and one for unknowns. And then we can also develop metabolome atlases of certain things that are apparent in certain regions of the brain, of the species, for example. So LC-Binbase, therefore, is powered automatically by massbank.us. We collaborate with massbank.japan and massbank.eu. So with the idea that when I go bankrupt, that at least the data are also saved either in Japan or in Europe. First, it was developed by Sajan Mehta, now Nolan Guzman. And we see that it's growing, and you can upload anything you like, but it has to have a structure with it. We do not allow unknowns in it. This is just for annotated compounds and MSMs. So with that input, we did the same analysis as before. We ask again, like, how can we do it in an automatic flow into LC-Binbase? And that's then the front end of LC-Binbase, how it looks like. So you would have, for example, here, the spectrum, the MSMS, and then manually you can annotate, okay, it is this following ceramide. And if you can add comments, and other users may disagree, and you can promote or demote annotations. So it's a little bit like Wikipedia, you know, so far within our lab, but of course we can open it to the rest of the world so that other people might disagree, and we would have an exchange of what this spectrum really means and how far we can go with the annotation. Then we have unknowns. Here's an unknown spectrum. Well, I don't know what it is, but you know, it's a mass 935 at normalized retention index 676. Well, if you have an idea, let's tell us. I mean, that's would be nice to know. And when we open up to the world, then you could do it. Now we can look into novel unknowns like oxidized triacylglycerides, for example. You know, and this specific one is high abundant in the heart. It's also found in muscle, but it's basically not found anywhere else. Another one had very similar patterns. And if we compare this to their corresponding native triacylglyceride, we see that, you know, the pattern is different. So for example, the native triacylglyceride would also be found in liver, but not in the brain, but it would not be oxidized. So we think that these oxidations, therefore, are indeed biochemical processes and not just mishandling of samples. We cannot completely say it's definitely not mishandling, but who knows? I mean, it doesn't, at least from the biology part, like comparing the oxidized to the native, it looks like this is real biochemistry. We see similar things in phosphatidylcholines. Again, the oxidized phosphatidylcholines, for example, very abundant in plasma, the native phosphatidylcholines not so abundant in plasma, and vice versa. Oxidized PC, not so abundant in brain, but the native, very abundant in brain. So again, giving us the idea that this is a PC with 3 oxygens on top of it. You know, the MSMS fits, but the precursor mass is clearly saying, no, it has 3 more oxygens somewhere. So it's kind of like a semi-annotation, if you like. And then we can look also on really annotated compounds, like just to give you a clue, you know, a PC 4422, I draw here versions of different isomers. This one was found all the time in the brain, basically nowhere else. Similarly, this hexosylceramide in the brain and in the muscle, but not in plasma, heart, or liver. We also see absence sometimes, like, for example, cholate and glycodeoxycholate were found in plasma and liver, the cholate also in the heart, but not in the brain or the muscle. And these ether lipids were not found in the heart or the liver, but they were very frequently found in brain, plasma, and muscle. So that gives us the idea we can go to atlases to kind of know for both knowns and unknowns where they are. How do we remove false positives, especially for unknowns? Well, when we have an idea of a structure, we can do machine learning and then say, well, you know, the expected retention time for this unknown should be here. And when we apply that, we are able to discard false positives to some extent. For example, without retention time prediction and really good MS-MS similarity, in the positive mode Q executive, we would have about 1900 annotations. And when we then exclude it based on retention time windows, we get only 1500. So we increase the confidence that a possible structure might be even what it is. We can, of course, go deeper. This is just, you know, for those of you who don't know, this is where we want to go. Deeper lipid identifications, including double bond positions, for example, using HCD and ultraviolet photodissociations at 220 nanometers to get the double bond positions right. That's a collaboration with Thermo Fisher on standards that we did. And it actually, many people published that before. We just confirmed that it's also working on this 220 nanometer position with these new instruments, both for the double bond positions, but also for the SN positions. So we can clearly say, you know, what kind of lipid that would be. So that works. And when we want to apply this whole idea, and we published that in December, we can now look much deeper into, you know, specific organs, for example, the brain. So we asked the question, how does the brain, mouse brain change during aging? So here we had 1500 identified metabolites, not only lipids, but also biogenic amines and primary metabolites. And we see that, for example, this specific diacylglyceride would increase over aging in all organs, but it would be at any time most dominant in the brainstem, that is the pons and medulla. So that's fine. So that's one lipid, but we can, of course, look at many, many other lipids. And some lipids show similar trends, like this ceramide would also have massive increase during aging, basically absent in adolescent in the different regions, but also much more present in the brainstem and in the midbrain. Whereas, you know, other lipids, for example, this specific ether lipid would only be found in the olfactory bulb and nowhere else at any given time. So it's really a specific lipid for a specific brain region. You know, we find, of course, lipids that are specific for organelles, like this BMP lipid. It's only found in lysosomes, so it's not present in any other organelle. So we can even go towards asking the question, like, how is the lysosome content changing perhaps over aging? And we see many, many other quite interesting lipids. This data is public. You can go to mouse.atlas.lipidomics.us and search for your favorite lipid and see what it's doing over aging. That's why we made data public. So we see triacylglycerides, they change by region, but not so much by age. Ceramides are very specific. Some of them decrease in aging. Some would stay the same, but have regio-specific, again, here in olfactory bulb. Similarly for ethanolamines, some of them would not change whatsoever, either by region or age. Others would have a huge decrease during aging. Similar for PCs and also for many other classes like monoisoglycerides. It showed a very different pattern for arachidonyl glycerol compared to monoolein. Cholesterols, specific cholesterol sulfate or cholesterol esters, much more abundant in the olfactory bulb, very different to free cholesterol. Sphingomyelins and gangliosides, dihydroceramides, all of them quite interesting to see what they do. That doesn't mean we know why they do it. It's a map, it's an atlas. It gives us some clue where to start, where to look, and also in kind of expectation when other people now do brain lipidomics in terms of aging. So overall, I'd like to conclude. You know, the NIH Metabolomics Consortium is now in its final year. Thanks for Charles Evans, who spearheaded that effort in this interlab comparison, and Tong Shen, who actually did a lot of data processing for that effort in my lab. So these two are the heroes of many of the slides even that I presented today. I've shown you that this worked because of standardization of data acquisitions and standardizations of data processing and output. We think internal standards are key for retention normalizations. And we are able to now enumerate at least the common core and specifics of unknowns and known lipids, where the future is to do it all through LC-Binbase and then apply it to atlases to define healthy and disease states. Thank you so much. And now I'm happy to entertain questions.
Thank you, Oliver. Thank you. That was great. Yeah, I'm really interested to see this, work to try and identify unknowns. It was something we had to go at a number of years ago in our lab and concluded it was just too difficult. But I think there's a couple of questions I have about this before we go further, if that's okay. I'll take chair's action. So yeah, it's a really challenging area, but I think it's really important to try and identify these unknowns because they could turn out to be biomarkers or metabolically important or anything. So it's quite pioneering work. Some of the cleanup, I mean, like when we try to do this, the big challenge was the cleanup. I think particularly of MS data, we would start with our Orbitrap with something like 60,000 features and then have to do all this processing to try and clean them up. And you mentioned a little bit of the strategies you have, but I think there's many more. And I wondered, did you deal with things like in-source fragments, common contaminants, salt clusters, isotopes, adduct duplicates, where one lipid shows up multiple times? I mean, did you have programs and algorithms to deal with all that too, or?
Yes, so especially in Michigan, they published quite a bit, like Binner and others.
Yeah.
So these can easily be applied on top of LC-Binbase. So we first say, if you have, say, a sodium adduct, then it definitely has a different accurate mass, and it does certainly have often a different MSMS. At first, it would be two different bits, right?
Yeah.
But they have the same retention time. And it's very easy to say, of course, they have the same retention time, right?
Yeah.
So, of course, it would be very, very simple to say, well, obviously, you're the sodium adduct, and obviously, you are the ammonium adduct, and whatnot. And we would combine them on that level. So we keep them as different entities in Binbase, but then we combine them, for example, in exports for the researchers so that the researcher only get maybe the combined value.
Yeah. So like when you show those Venn diagrams, you've got rid of all of that stuff at that point. No, I hope you have.
Okay. So you got to think about, we are regular humans, right? So we only have that much time, and it's not the only project we're working on. Mm-hmm. So that one was really still mostly on the level of, here we are. Charles would know better, really, how much he applied his own software for it.
Yeah.
And we did a lot of cleanup, but maybe not to the extent that I would say it's 1527.
Yeah, it's difficult. It is. Yeah, it's difficult.
But the overall numbers are okay. And more importantly, also the ratios. What I found is that, for example, the unknowns are often specific. So that's interesting. Mm-hmm. I always suspected that. Right? Mm-hmm. So there are specific lipids in, say, the brain or in, and then I showed you also there are specific lipids in specific brain regions at specific aging times. That's kind of cool. Yeah, it is. Right? Yeah. So, it's kind of like showing that there is biology to be learned.
Yeah, absolutely. I totally agree with that. It's just, it is challenging. So another question, you mentioned a few times you were finding oxidized lipids, and you showed some triglycerides, and then there was also a PE that was a 16-0 with 3 oxygens. And I did a couple of questions about that because, to have the triglycerides seem to have like one double bond in the total, all the fatty acids. And I think how you would oxidize that in terms of the usual kind of enzymatic or Fenton chemistry type oxygenations, I'm just not sure that we would see much of that. It should be more the kind of polyunsaturates that are oxidized. And 16-0 with 3 oxygens, it flashed past me very quickly, so I didn't get to see the accurate mass, but it showed up as 303 with a 766 precursor, which is quite typical of arachidonic acid in a PE as well. So yeah,
I know.
I'm not sure about
I
three
agree.
oxygens and
So
6
right now,
double bonds.
it is just, that's why we call it an annotation and not an identification. So
Yeah.
we wouldn't know where it is, and it obviously must be some form of epoxide or something like that, right?
Yeah.
Could it sit somewhere else? I don't know. It is too many things to investigate.
Yeah.
And right now, you know, that's the idea of a database approach. You would say, it's a feature, it's a thing. We see it all the time. Let's find out how it's done, right?
Yeah, and if it's
important,
important,
whenever I thought I knew about lipids, then I was just turn around the corner, and then you're like, huh, there's another thing I never heard about.
Well, yeah, I know. There's so much to learn. Okay, so I should stop monopolizing the question and answers and pass you over to some of our speakers. So the first question is from Hong Yin, who you should be able to unmute yourself now, Hong. Are you able to speak?
Hong?
If not, we'll come back to Hong. And okay, Hong is there. Go ahead.
Oh, hi, Oliver. What a wonderful presentation. I was just curious, I mean, just tapping into your vast knowledge base between the, you know, the structure serves function. This is a fundamental biological maxiom. So it shouldn't be surprising to see the brains has their specific lipidome and so on and so forth. So among the lipidome, metabolome, and the proteome between the organs, which of the has the largest differences?
We will be able to tell you that in 10 years, hopefully.
Okay.
I don't think we're there yet. I don't think so.
But are you connected with
the biggest complexity is in the GI tract, because you have all the microbes there. Mm. If you want to look into organs, that's where the beauty of where the intersection of human genes, microbial genes and enzymes, plus food and exposome meets. That's like the New York of organs is the gut intestinal tract. Kind of fun. Hard to
well, not necessarily in a epidemiological sense of a scale. You know, don't have to looking at the population, but a single human being or a single organism like a mouse, which is raised up in a standard condition. How are they comparing between organs? I'm not concerning of a human population.
I don't, honestly. We need atlases for that. And to some extent, you can get it from the literature by text mining. That is, for example, the Human Metabolome Database from David Wishart. We did similar things on just blood, but you don't find it across all organs. You know, what makes the pancreas specific? You don't have that information on databases. Why? Well, because nobody funded it. Usually get funded to fight a certain disease. That's where at least the NIH, and I think also in Europe, you know, like fight cancer this and fight cancer that. That's fine, and we do our database stuff on the side, which is a bit sad, but that's where we are.
Okay, thank you. So Ali Hajaya, you should be able to unmute yourself.
Hello, great talk. I'm always skeptical of lipid extraction methods and what makes it there. I don't know if you know if anybody has done any proteomic analysis on lipid extracts.
So first of all, you're right. If you have just an 80% methanol crash, you will find a lot of proteins in that. So that would not be a 100% proteins in the pellet. You're absolutely right. You will, in classic ways of looking at extracts, you will find proteins and residuals. And what we found is when you dry down and reconstitute, the protein fraction would not usually reconstitute, whereas your metabolites or lipids would. Secondly, you can also do it by a re-extraction to get really to 100% protein collapsing and nothing in the lipidome/metabolome fractions. We, in our case here, we use a modified Matthias extraction with MTBE and methanol water. Some other people use different ways of separating polar compounds to lipids. But yeah, you're right. I mean, every extraction has its own biases and pitfalls.
Thank you. I have another question, if you don't mind.
Yeah, go ahead.
You mentioned building a database. Does that only include plasma and tissue, or do you also take cell culture lipid extracts?
Yes. So in GC-Binbase, we have all that. So we have, I don't know, over 100 species and 200 organs in GC-Binbase, which is available under binvestigate.fiennlab.ucdavis.edu. You know, but it's just GC. It's kind of like a little, you know, like a small thing. And so we want to do the same for LC. That's called LC-Binbase. And then we will have the ability to say, well, this is the so-and-so like fibroblast, and this is E. coli, and you name it, right?
Thank you.
Mm-hmm.
Okay, so Christina Ferreira, you can unmute yourself.
Yeah, can you hear me?
Yeah.
Yeah, thank you so much for this opportunity, Valerie. Thank you for your amazing talk, Dr. Fiehn. I was wondering how much the chromatography system may have biased the results to, you know, that lipids have a large polarity range. And so this may be a filter for that.
Yeah, so we use, okay, so this is not an advertisement, but we use Waters columns. They are the best. First of all. Secondly, we tended to use the charged surface hybrid C18 column. I think even for this talk, I only talked about CSH, but now we actually switched to bridged ethylene hybrid columns, C18, which are a little bit broader, also better for phosphatidic acids. So the CSH column had a little bias against phosphatidic acids, whereas the BEH is a little worse in the actual chromatography, but it allows nice retention of phosphatic acids. So yeah, but hey, there's many other columns. Be my guest.
Okay, thank you. I have a second question.
Quickly, go on, yeah.
Yeah, if you could comment a bit more on the oxidized lipids that are very much in the interest of many people, because they were detected in the negative ion mode and the non-oxidized in the positive ion mode. If you could comment on that.
These are unknowns. We don't know yet what we are really talking about. These are clear MS-MS similarities with different acrid masses, and I think that's where we are. We didn't do a lot more investigations in where the oxygen sits. Would be nice. I mean, for some, it's known. Obviously, you would have close to a double bond, as Valerie said, usually, or adjacent, actually. So what we did notice when we have things that are occurring in both positive and negative mode, they usually correlate very nicely. So I don't think we have a huge bias in terms of, you know, either detecting only things in positive or only in negative mode.
Okay.
And oxidized lipids can be, oxidized triglycerides would be, of course, found in positive mode, obviously.
Mm-hmm.
Okay, thank you, Christina. So, Jose, are you there?
Yep.
You're
Okay.
unmute yourself. Yep.
Hi.
Yep.
Yeah.
Hear me?
Yep.
Thank you. I just wonder about if have analyzed stool.
Yes.
And how hard is to analyze stool, because I think it's a tricky biosample.
So stool. Stool has much fewer lipid contents than other organs. So in terms of lipidomics, stool is the not so interesting. Maybe bacterial lipids, but not the other, you know, overall, because we take out the energy from the stool and put it to our own uses. The upper GI tract is more interesting in lipids. I suspect that FAFA lipids are made in the upper GI tract and then absorbed into the humans. So that's kind of cool. We looked into the upper GI tract in humans. We have a paper in review in Nature Nature currently. We'll see about what that does. We'll submit more papers on that. So yeah, stool is kind of very diverse, not so much in lipids.
Okay, but in general, what type of lipids you can find in stool? Can you find like also the rocks?
Regular types.
Okay.
Do I see PIPs, phosphoinositides or phosphoinositide I don't. I sometimes have too many acronyms. There was a chat asking. Okay. There's so many acronyms these days.
Okay.
Phosphate. Yeah, I see a lot of phosphates.
Okay.
So thank you. That is only my question. Thank you.
Okay, Samandla Changazi.
Yes, I'm here.
Hello, go for it.
Yes, thanks, Oliver, for the great talk. My question is a bit of a specific question. How do you normally deal with in-source fragmentation? I know Valerie already touched on that, like asked the similar question, but I just want to know the specific.
So
I know it differs.
First of all, it's a great question. Secondly, to put yourself out in the public is always in danger of, how can I say, embarrassing yourself. So I did embarrass myself proudly by making my data public, for example, in the ADNI consortium. So the ADNI consortium is Alzheimer's Disease Network Initiative, and all data are publicly available, so you can download. And then Peter Merkle from Australia, he's a really good lipidomics guy, downloaded my data and proved me wrong. He said, this lipid is an in-source fragmentation. You totally misannotated it. Yada, yada, you're a bad person. And I said, you're my friend, Peter. First of all, don't call me a bad person. And secondly, thank you for noting my error, and I will correct it. So that's, yes. We do this by retention time, basically modeling, right? So that's the idea of retention time modeling. If you have the wrong lipid at a wrong retention time, then it is an in-source fragmentation. That's one way to find it out.
Okay.
And there's other ways, like, changing your ionization energy and yada, yada. This has been published, not by my lab, by other labs.
Okay. Thank you. So we've got a few more questions and then we'll finish up. So Stella Adamowo has got, I thought it was 2 questions, but it's the same question posted in 2 places. So go ahead, Stella.
Sorry, thanks, Valerie. I thought you were monitoring the chat.
I know, I'm trying to go between 2 different things and making sure that I'm trying to take everyone in order, but go ahead.
Thank you. Yeah, thanks, Dr. Fiehn, for the talk. I was just wondering, I know you've said the oxidized lipids are unknown, but for you to have classified them as unknown, it means you've identified them as oxidized lipids. So by any chance, did you see more of these modified lipids, especially the oxidized lipids, in a particular organ compared to the others?
We're still looking at the data. We haven't published. This was, by the way, almost all unpublished data. So we are still working on it. It is hard, because you run out of time and your people want to graduate and you leave. At some point we'll cut it off, and then we put all the data out there for you to investigate. So I cannot give you a good answer on that, because simply put, we're still ongoing in terms of data processing and annotations and arguing. This is a consortium. It's not just me, but it's also Charles and others. And we do it on the side. It's not like the main project we work on. Usually the main project is like pain, chronic pain, or longevity, or you name it, right? These are my things I'm funded for, not for hunting lipids across organs.
But those are projects that aren't involving lipids, is that right? Is that why you didn't talk about them
Yeah,
today?
no, man, these are projects that are even less public.
Okay.
Thank you.
Thank you, Stella. So I think we should probably take one more because you've got loads of questions, which is just fantastic, but we are like already 5 minutes over. So Joshua Morris, you should be able to unmute yourself.
Hello. Hello, can you all hear me?
Yeah, we can hear you.
All right. Thank you, Oliver. Fantastic talk. So I just wanted to hear briefly if you could comment on the chemoinformatic strategies that you use to remove any of those false positives that you identified.
Well, I think retention time prediction is something that has been undervalued in the community. A retention time is a hugely important character in a good chromatography and a standardized chromatography. So we work here with Paolo Ponini, and we published last year or the year before a combination of different informatic schemes, machine learning tools from Keras to neural networks, and of course, random forest and others. And this year we will publish RETIP 2.0, like retention time prediction 2.0, which will work on AutoGluon. AutoGluon is a framework of machine learning software that basically picks the winner. It will be very hard to beat AutoGluon. It's been released by Google maybe a year ago, and it's very successful, and it improves the retention time prediction even further, specifically for lipidomics. So lipidomics is really, these things can be fairly nicely predicted. And, that's where we think, and in addition, of course, I didn't talk about ion mobility, other people do. The idea is then you put on the ion mobility or the collision cross-section on top of that. We didn't do that because at that time, we just didn't have an ion mobility mass spectrometer, but now we do. And, that gives us will give us another dimension, or the community another dimension to go deeper. So these are the chem informatics tools that have been underappreciated, I think, over time.
Thank you.
Oh, we also have a new, I didn't say, we have a new MS similarity code that we published in Nature Methods in December called Spectral Entropy Similarity. Read about that. That's also cool.
Okay.
Appreciate it. Thank you.
Okay, thank you, Josh. So I think in the interest of time, we should probably end. So I just want to say thank you very much for Oliver. Fantastic talk. Great to hear the update about the work and the informatics in particular. And thank you everybody for coming today. And our next webinar, which is on the 17th of May, is Michael Holchipek talking about profiling of human serum for detection of pancreatic cancer. So hope to see you all then. Thank you very much.
Thank you. Bye-bye.